This meeting is now being recorded.
Well, good evening. Welcome to the EDS Awareness Educational Series webinar. My name is John Furman. Before we introduce our speaker this evening, I'd like to give you an overview of our free program for those attending for the first time. So we'll go over who we are, what is our program, some upcoming events, introduce our speaker for this evening, and then there'll be Q&A. So again, my name is John Furman. I manage the National EDS Awareness and Chronic Pain Partners nonprofit organization based in Cincinnati, Ohio. My daughter, Deanna, who's also on the call, leads the Cleveland, Ohio EDS support. She was diagnosed with hypermobile EDS in 2008, the same year my wife, Carol, passed away with breast cancer. I was a caregiver for my wife who struggled with undiagnosed EDS for over 30 years. So we introduced our program at the 2012 EDS conference to help EDSers form independent local support groups and spread EDS awareness in their communities. We've been a sponsor for these conferences for the last 5 years. We started over 115 groups to date. Each group is given their own free website with a link to the directory and map. We receive feedback that conferences provide valuable information and social opportunities that many cannot physically or financially afford to attend in person. So we decided to bring the speakers to you through the free EDS Awareness Educational Series. We typically meet monthly at 7:30 Eastern Daylight Time. All the programs are free, the meeting announcements, and whenever possible, the webinar recordings will be posted on our site at edsawareness.com for later replay. You may receive email announcements for future sessions by requesting the free guide on our site. Tonight's presentation is sponsored by Body Support Score, where you will find over 250 products selected by EDSers for EDSers. Proceeds from our store and the donations to our nonprofit organization help fund these free programs. Please visit our store and check out the helpful products that we offer. So just a general disclaimer, this presentation contains general information about EDS. Members of the EDS community voluntarily participate in this program. The information is not advice. If you're having medical problems, now, please call 911 or the emergency services in your location. Always consult your doctor before making any changes to your treatment.
Our webinar for next month will be on Thursday, September 19. Dr. Ann Maitland will be returning for the third time to discuss mast cell activation updates. To view upcoming speakers, always visit the webinars page at edsawareness.com for the full schedule. For those attending live, there will be an opportunity for Q&A after this presentation. Please add your questions at any time by clicking the Q&A icon at the top of your screen. After typing your question, click the orange button to submit. Our speaker tonight is Dr. Joshua Milner, and he is going to talk about Tripped Up by Tryptase. He did his undergraduate in biology from the Massachusetts Institute of Technology, MIT, in 1995 and completed his MD with distinction in immunology from the Albert Einstein College of Medicine. Dr. Milner completed his residency in pediatrics at the Children's National Medical Center in Washington, D.C. He was a recipient of the Pediatric Scientist Development Program Fellowship and did his fellowship in allergy and immunology at the NIH. He completed a postdoctoral fellowship with Dr. William E. Paul, examining issues of mouse T-cell receptor repertoires before beginning as a clinical tenure track investigator at the LAD. And he was named chief at the LAD in 2017. He is an elected member of the American Society for Clinical Investigation and the Association of American Physicians. He is the recipient of the Feta Allergy Research Forum Award, as well as the Drunkier Prize in Pediatric Research from Cornell University. And it is our great pleasure to welcome Dr. Milner to speak to us tonight. And thank you, Dr. Milner, for your time and sharing your research with us. I'm going to go ahead and turn over the microphone to you for your presentation.
Well, thank you very much for the opportunity to chat to everyone. Everyone looks like a beautiful number right now, but I'm sure that behind that are great faces. I'm usually used to speaking to live audiences that I can see. So if I can't interact with you the same way that I usually do, I'm going to try. But anyway, so I wanted to talk about what we've been doing at the NIH with respect to tryptase. I'll start just by giving a brief introduction to the allergic response and then get into what it is that we're doing and why that we were doing it. So just to get everybody on the same page, we have a lot of antibodies floating around in our body that are there usually to grab foreign objects to stop us from getting infected. And there are different types of antibodies, IgM, IgG, IgA, and a special one called IgE. And IgE is of particular interest because what it does is it actually binds to something called a mast cell, which I'm sure you've heard of. The mast cell is not a disease. It's a cell that's in your body. And we'll get back to that in a little bit. It's coded with a receptor called FcεRI1. And it has inside of it histamine and other stuff that can cause all sorts of havoc when that mast cell releases what's inside of it. So IgE is usually sitting on the surface of FcεRI1, and if you happen to have IgE that's specific for something like a peanut, and I can assure you this is not drawn to scale, if it's specific for the peanut, then that IgE will actually turn the mast cell on. Lightning bolts literally fly out of the surface of the mast cell, and it pushes what's inside out, like histamine and other things. And that, depending on where it's acting, like your skin or your gut or your nasal mucosa or who knows where else, it will cause all sorts of trouble. And the main thing that it does is it dilates your vessels, and it releases different types of chemicals that can cause itch, that can cause redness, that can cause inflammation, a very specific type of inflammation. So you can get leaky blood vessels, you can get local inflammation, and depending on where it is, again, you can get hives, itchy skin, sneezing, wheezing, runny nose, vomiting, and unfortunately, should it happen throughout your entire body, you can actually get anaphylaxis, because if your blood vessels leak too much or become too dilated, which is one of the other things that histamine can do, amongst other things in the mast cells, then you can't maintain your blood pressure, and your blood pressure drops. And that's what anaphylaxis is. Now, there are other ways to get your blood pressure dropping that aren't anaphylaxis, and sometimes that can end up being confusing. One thing that we do know is that there are people who are out there whose mast cells can get activated, it seems, even without having the IgE bind something. It can just happen out of nowhere. As a pretty interesting example, you actually can have certain patients who are allergic to cold, and cold alone will make the mast cell degranulate. Other people can have vibration. Believe it or not, there's a gene that vibration can cause their mast cells to degranulate, and so there may be people who are listening right now who recognize that, like, let's say if they were mowing the lawn or they're using a hand mixer or something like that, they can actually get itchy, that is something that can happen. Sometimes it's genetic, and sometimes we don't know the reason why that ends up happening. But the bottom line is that the mast cell can be activated either by something external or in some cases there's just something about it that makes it very fragile and it will release its contents for all sorts of different reasons. So my lab is mostly interested in trying to find genetic reasons for allergic problems, and we tend to focus on folks who have either severe or very unique types of allergic issues, usually when it runs in the family, and often when it's associated with something besides just the allergy, because that's our clue that more than one thing, that there might be something more complicated going on, that it could be genetic. That being said, two things can happen to all sorts of people, and that's an important point to make. But we were referred an 11-year-old girl who had lots of recurrent episodes of her face getting very flush. She had lots of episodes of abdominal pain. She would get headaches. Pruritus means itching. She'd already had her gallbladder taken out, which is what a cholecystectomy is, for biliary dyskinesia and chronic abdominal pain. Unfortunately, the chronic abdominal pain didn't get better, even though you could see biliary dyskinesia on her studies. So they tried the surgery and it didn't work. She did have hypermobile joints. She also had tachycardia and episodes of syncope, and she had a diagnosis of POTS. Her basal serum tryptase level, which I'll explain in a second, was high at 22, and that prompted their doctor to look for something called mastocytosis, which I'll also explain in a second. And lo and behold, even though they expected with someone with these kinds of symptoms and a high tryptase to have an abnormal bone marrow, she did not. Her bone marrow was completely normal. But then when you talk to the rest of the family, her mom, her sister, her brother, they all had their blood drawn for different reasons and found their tryptase to be high at baseline as well. They had some flushing. They had some abdominal pain. They even eventually at a certain point in time got diagnosed with POTS. But they said, it's not as bad as whatever it is that she has. And so they didn't really, pay as much attention to the fact that it was in the rest of the family. And in fact, Mom was pretty clear that she said she really never had any problems until the second child was born. And then after that event, she started having all of these symptoms. And she also had such bad abdominal pain that she was thinking of getting her gallbladder taken out, even though it didn't really work for her daughter. All of them were diagnosed with what was then called Ehlers-Danlos syndrome type 3, or what we know now as hypermobile Ehlers-Danlos as well. And the question was, does any of this have to do does one thing have to do with the other? So after meeting them, we met a bunch of other families with a very, very similar type of a story. And so these are just pedigrees of males are square and females are circles. It's not a judgment. It's just what someone decided to do as a convention for drawing what a pedigree looks like. And as you can see, it's in every generation that you have people who are affected. They're the ones who are colored in, who are darkened in, whereas the ones who are not affected are the white boxes. And they're in every generation, and there could be multiple people affected, boys and girls, in each generation, and that means it's an autosomal dominant inheritance of having a high tryptase, which wasn't, as of about 5 years ago, no one had ever even seen such a thing. There was no such thing that you would have a high tryptase running in the family. It hadn't been checked. People hadn't thought to look in multiple family members. This just shows you some of the things they can get. This actually is a patient. Some of these folks definitely have issues with vibration causing their mast cells to degranulate. This person's arm, we put it on a vortex, which normally shakes up test tubes, but here we actually do it on patients' arms because we're mean at the NIH. What you can see is actually a hive almost here, but then redness and itching, which extends well beyond where the vibration occurred on the arm. This just is an example of the hypermobility, which I'm sure a lot of the folks in this audience, it's not something new for them. When we collected more and more and more information on these families, we saw the following things. There were people who would have systemic reactions, often a number of them to like bee venom or hornet or wasp venom. Those who had flushing and itching and associated sleep disruption, some who had hives actually that chronically. There were definitely connective tissue abnormalities. In addition to the hypermobility, they had their primary teeth had to be pulled by a dentist, and they also had chronic pain. And then there were those who had functional GI disorders, what were called functional GI disorders, such as irritable bowel syndrome, chronic reflux symptoms, but where when they would get scoped, there wouldn't be anything to find, even though they had very, very substantial complaints. Poor motility in the GI tract also was noted by a lot of folks, difficulty swallowing, all the way down to really terrible constipation and things like that. A variety of different GI complaints. And then quite a number of folks had issues with dysautonomia of all sorts of different, and here I'm saying dysautonomia in the sense that the body had very difficult time keeping blood pressure and pulse, and even the dilation of the blood vessels under control. So there could be lots and lots of different ways that this would be manifest. And in fact, we don't know in many cases if flushing is because of mast cells degranulating or if it's because of an underlying problem with dysautonomia, with the nerves and the blood vessels not talking to each other in the right way and not being able to be controlled as tightly as they usually are. So when we saw those initially we just saw maybe about 10 families, and since we've seen 50, 60 or plus families and hundreds of folks who are affected, we thought, well, that's really interesting because what was being thought about in a lot of these patients were, and the reason why they were sent to the NIH, which is important because that refers to something called referral bias, where the reason they were sent makes it look like a lot of people have one thing, but that's because that's why they're being sent. They're being sent because of itching, flushing, rapid GI pain, headache, loss of blood pressure, and anaphylaxis. Those are things you might see in a rare disorder called mastocytosis, which I'm going to explain a bit more. And so the fact that we saw that in a lot of our patients reflects the fact that that's who gets sent to the NIH often is because someone thinks that that's what's going on. Whether that reflects what was going on in these families broadly is very hard to know because we only got the folks who were sent to us. And so that was the symptom that was going on. And on the other side, we know that there are folks who have hypermobile EDS, joint hypermobility. Some of them have retained primary teeth, scoliosis, pectus, a number of different skeletal issues that can end up happening. But the two of them together, if you look at them carefully, actually can share a number of symptoms like dysautonomia or POTS, or abdominal pain and IBS, or dysmotility, or chronic pain of all sorts. Something called brain fog, where it's just really hard to concentrate, and it's not just because you're tired or depressed or something like that. Easy bruising and bleeding, which we certainly see in both cases, which is interesting, and not completely clear as to why. And a bunch of neurocognitive and neuropsychiatric issues that can be shared. And we don't know whether one drives the other, they're both shared, or whether this is something that happens. For instance, we've certainly seen those types of symptoms, oddly, in lots of rare chronic diseases that we see at the NIH. So again, because we're at the NIH, we get referred tons of extremely rare disorder, whether it's a genetic abnormality that's been identified and it might affect your kidneys or your heart or something like that. And over time, we've noticed that there could be folks who have very, very unrelated rare genetic disorders who at the same time might also end up having a good number of these symptoms that are in the middle of this Venn diagram that I've drawn, which makes it all the more complicated when we try to understand what's happening. Is it something intrinsic to the disease, or is it having that other disease for such a long time starts to trigger a common thing for even though it started in a completely different way, and we really don't understand that super, super well, and it merits a lot of future study. So mastocytosis is essentially abnormal growth of mast cells in your bone marrow and in other tissues. And it's rare, very rare. Probably 1 in 100,000 people end up having mastocytosis. The ways that it can be very clear is a rash called urticaria pigmentosa, which is almost a large freckle rash that when you scratch it, it actually hives up. You can also have episodes of flushing, like this woman has here, itching, severe abdominal pain. You also can have just idiopathic anaphylaxis, where out of the blue you pass out and might have broken out into hives and your throat closed. And then also some bone issues, osteopenia. It's associated very often with having a high basal serum tryptase, which is why the clinician thought that maybe the girl that I originally presented had mastocytosis. But it's clearly known that in most cases, and definitely not all, most cases of mastocytosis are caused by a mutation in a growth factor receptor on a mast cell. That which keeps it dividing and growing is something called KIT, and there's a mutation that people can get, not that they're born with, but that happens in their bone marrow the way, unfortunately, cancers happen, where this mutation called DH16V, and there are others that have been found as well, turn on this growth factor receptor, and it just makes the mast cells grow and grow and grow, and really they're not regulated, and they start depositing in the tissues and causing all the different symptoms that you see. There can be triggers, but it also can just sort of happen out of nowhere where you get these symptoms because you have tons and tons of mast cells all throughout, and that can be in different parts, in different organs and that sort of a thing. Tryptase is one of the things inside of the mast cell. So when I showed you before that histamine can be inside of these granules, tryptase is another protein that can be found inside of these granules. And tryptase is usually released as actually it's called a tetramer, or 4 pieces of tryptase. But sometimes tryptase can also leak out as what's called a monomer, or 1 piece. And the leaking is what causes the regular, everybody just about has a measurable serum tryptase, and that's mostly thought to be leaking out of these vesicles that are here inside of the mast cell. Either alpha or beta tryptase are just leaking out. They're not being thrust out like when a mast cell degranulates, has been activated, and you get these tetramers. And we sort of have an idea of what this does, these tetramers here, but we don't really know what these monomers are doing floating around when you have a basal serum tryptase, which makes it hard to know exactly what it's doing. It's a mast cell product. It's usually released during IgE-mediated allergic reactions. There are some rare blood cancers that can be associated with having a high tryptase. Mastocytosis, of course, is the main one that we usually associate with having lots of tryptase in the periphery, and that's because simply we have tons and tons of mast cells, and they're leaking. They're going to end up with more and more tryptase floating around. And then there are folks who have mast cell-associated disorders that aren't due to mastocytosis. Some people, they've been termed mast cell activation syndromes, but we don't really have a great handle on those. And I know that Dr. Maitland, with whom I've often worked closely, I know that she'll be speaking about this next, and we'll go into it in even greater detail. But the important point here, from the point of view of someone studying this, is that we don't have a great handle at all on what exactly mast cell activation syndrome is. There are lots of folks who have very similar symptoms, but we don't really know if it's one thing, if it's 20 things, if there are different forms of it. But the bottom line is it tends to, for the most part, almost by definition, is it's not mastocytosis. You don't have tons of mast cells, but those mast cells which are around are causing lots and lots of trouble, possibly. It's mostly a clinical diagnosis. There are labs that can be drawn, but I have tons of patients who don't have a single lab wrong with them and look exactly and precisely the same as someone who's gotten $1 million workup and gotten every test run. And really, in the end, I can't tell the difference between the two patients, to be honest. It's mostly how they respond to treatment and not what tests they get done that helps me determine what types of disease the patient has. Now, the thing that has been known for a while that people just did not really pay much attention to, mostly because it was just a statistical way that we decided that tryptase is high. We said, all right, let's just cut it off. The top 5% of tryptase is going to be right here at 11.4 micrograms per liter, because statistically that's a number we do, or top 4.3%. And literally, that's what we use to define what a high tryptase is. It's not because suddenly we found all these sick patients with a tryptase of 11.5. It's just statistically we decided that's what high is. So it's not that if you have 11.3, you're fine, and if you have 11.5, you're not. It's not that. It's just was defined statistically. And patients who have mastocytosis, for the record, can have tryptases way up here in the hundreds, even in some cases. that's when you can be fairly sure that it's pretty bad. When you're down in this range, this was a bunch of normal volunteers who volunteered for studies, and in one study after the next, it's usually about the same, that about 4% of the patients have a tryptase higher than 11.4. But again, that was just a statistical cutoff. It wasn't a disease-defined cutoff. It was a couple of years ago, very interestingly, in a journal, it wasn't really noticed by too many folks, a very large allergy practice in Austria drew tryptase levels on 15,000 patients who came in through their door, and to get a real sense of how high tryptases could be and whether having the high tryptase had anything to do with the symptoms that the patients came in with. Now, again, there's referral bias because these patients are coming into an allergy practice, and the symptoms were mostly from patient reports, so there was no way to sort of independently validate the symptoms, but these were just complaints that the patients reported. And what's pretty amazing is that the patients certainly did not know their tryptase walking in, and neither did the docs. But when the docs analyzed the data and looked and compared, let's look at people who have a tryptase higher than 11 and compare them to the people who have a tryptase lower than 11, what symptoms are different in the ones who have tryptase higher than 11? And it actually didn't matter if it was higher than 11, higher than 15, higher than 20, or more. It just had to be higher. And what they found was that there was flushing and angioedema, which is swelling that you can get, that was much higher in those who had a high tryptase than not. Gas, diarrhea, and abdominal pain, the lovely gastrointestinal symptoms that can happen, very similar to what we saw in the patients who were originally referred to us. Vertigo, tachycardia, palpitations, and even hypotension, again, significantly higher just for having a high tryptase. Oops. If you notice, number one is fatigue. Over half of the people who had a high tryptase had fatigue. But if you also notice, 37%, 1 in 3 people who walked through the door complained of fatigue with a tryptase lower than 11.4. If you notice, a lot of these complaints were very common in folks. We don't know, though, for instance, how many people had, like, everything on the list, or 10 of the things on the list. How many was it happening at the same time? The important point was that it did seem that a lot of the symptoms that we saw in the original folks that we've been following who we thought had a rare disease, you could find that in anybody who had a tryptase more than 11.4. Okay. Now, it's just important to point out that you can get flushing, tachycardia, loss of blood pressure, belly pain, and even hives from a lot of different sources, okay? So you can get hives from eating a peanut, but actually the number one way that you can get hives is stress. I think most folks know someone who broke out into hives after something stressful, or just before a final exam or something like that. That is the number one way we tend to find hives in sort of all comers in the patients. It's rare that doing a skin prick test is going to find a reason beyond that. There's also hormonal reasons, thyroid issues, menopause, autonomic reasons, issues with the nerves and blood vessels talking to each other, including the heart, such as dysautonomia or something called idiopathic dysautonomia, where we don't know what's causing it. Is what's causing it mast cell problems? We just don't know. We really don't know that yet, although treating some of the folks who we've seen has helped some of their dysautonomia, but not everyone. And mast cells themselves can be contributing, but they themselves aren't necessary for most of the symptoms that the folks that we've seen have had. They're not necessary, but they can certainly cause it. It's important to point out that tryptase itself, usually when it's in that what we call the tetramer, or the four piece of a puzzle configuration when it gets released from the mast cells, but nonetheless, tryptase can act on something called PAR2, which is a receptor in the body, throughout the body. It's definitely in nerves, it's in blood vessels, it's in connective tissue, and it might even be on mast cells. And so tryptase itself might act on these different tissues and may well be able to contribute to lots of the problems without actually having to activate the mast cell. It's not clear. We don't know. But the tryptase could actually cause itself, could potentiate pain, could potentiate dilation of the blood vessels. By potentiate, I mean make it worse. So there may be a very typical thing that happens, but the fact that there's more tryptase there may make you respond to it more. Okay? It also is very important to point out that in addition to PAR2 acting in different ways, mast cells have on their surface stress hormone receptors, and for whatever reason, maybe just evolutionarily, it was important that mast cells get activated more easily when you were under stress. The bottom line is that the more stress hormones that are around, the easier it is for a mast cell to be activated. The other important thing, which is just more of a laboratory study, but which I think is very important, is that PAR2, this receptor that tryptase can act on throughout the body, you have to have it to get something called sickness behavior. So in mice, you can give mice a chronic infection, and in doing so, they stop eating regularly, they stop moving around, they become anhedonic, they don't enjoy things. It's distinct from being depressed or anxious or something like that. It's a way of the body shutting down in response to something like, let's say, an infection, so that it can do its thing I guess, and not have valuable resources used in different ways and not have you move around too much or whatever so that it can fight the infection. But sickness behavior can happen even artificially, and it seems like you have to have PAR2 to have that sickness behavior. And so you could imagine that there might be a number of physiologic changes that would happen if there was too much tryptase going around that might lead to some of the things that we saw in some of our patients. But I'm just explaining that as a way to understand what could be going on, but we obviously don't know yet. This is just in the research stage. Okay. I also love to show this study, which basically showed that when you have increased corticotropin-releasing hormone that happens from psychological stress, it can cause gut leakiness, actual leakiness of the gut, which is something that can be seen in irritable bowel syndrome. And actually treating with cromolyn sodium or gastrocrom, treating patients with that beforehand blocked the mast cells being activated by this corticotropin-releasing hormone when they got up to give a public talk in front of thousands of people, and remarkably, they didn't have the gastrointestinal issues when they beforehand blocked their mast cells, and it was amazing. They did not have any of these symptoms if they got shocked. So it's interesting that different types of stress lead to different types of programs in our bodies that can lead to different types of symptoms. It's actually interesting because we did notice, and lots of patients told us, that either they were fine or their symptoms weren't too bad until, I think as I mentioned with the woman at the very beginning, childbirth is what triggered her. For other folks, it could be a car wreck, it could be a divorce, it could be all sorts of different things. From then, they noticed that they started to have lots and lots of these symptoms. And so I think studies like this one, where you can show the way that a stressful event can actually, not a physically stressful event, but a psychologically stressful event, can actually trigger mast cells to degranulate. It's rather remarkable. And it is similar to what we've heard from some of our patients. Others born from the get-go with all of the issues. And it's very hard for us to understand exactly what triggers one and not the other earlier on. And the other thing we always have to worry about is something called recall bias, which is it really that stressful event that was the trigger for the symptoms, or were the symptoms sort of going on all the time, or with something else, and it's just that the stressful event is what you remember the most, and really what it could have been was an encounter with a butterfly that just wasn't a big deal. I mean, who knows? Please don't go out and avoid butterflies. It's just not going to help, I don't think, with most of the mast cell activation symptoms, but you get my drift. And so that's certainly something that merits us studying a whole lot more. Okay. So these symptoms, again, they may or may not due to mast cells being abnormally activated. Some folks, we did notice that they were making other mediators, and again, Dr. Maitland, I'm sure will talk about that a lot more, besides tryptase. They may have had something called an elevated PGD2 or a urinary N-methylhistamine. There are some other things that people are experimentally trying to figure out if that will tell you that mast cells are activated. But there were plenty of folks who had nothing else going on that would indicate that their mast cells are being abnormally activated. The mast cells were found in skin and in the gut. If a biopsy were done, in some cases there were more mast cells. They didn't look abnormal, but there were more of them. And in many cases, absolutely no more of them, nothing going on in the skin or in the gut, yet they were just as symptomatic. Okay? And so, but nonetheless, lots of folks did seem to respond to being treated with something that would either block their mast cells from releasing what's inside, and the medications that can do that are gastrocrom and ketotifen, or blocking what the mast cells make, such as antihistamines like cetirizine or fexofenadine, and also other types of antihistamines, which actually, like such as Zantac, ranitidine, actually can block histamine as well, and we usually add the two of them together first. Those and a couple of other, just a few other drugs that folks try, the folks can respond to those even though they're not hyper-producing things, their mast cells are not hyper-producing, at least that we can measure, and that well could be that because there's an overactive response to what a mast cell is normally releasing, as opposed to the mast cell suddenly releasing lots and lots of stuff, and then having a normal response to lots of stuff. So both may well end up be happening. We don't know in a lot of our patients. We assume when the laboratory is high that that's what's causing their symptoms, but that still remains to be seen for quite a lot of folks. So why were these families having high tryptase, and why might it be something important? Well, to make a really long story short, some amazing work that was done by John Lyons, who is a junior faculty at the NIH now in my lab, who's also an allergist and immunologist. The gene that actually makes tryptase is a really complicated gene, and this is just a schematic diagram of what it could look like on a chromosome. The weird thing is that there are 4 different places where tryptase gets made. I guess in the red, we'll say that that's where beta tryptase gets made. In the purple, that's where something called gamma tryptase gets made. And there's even another beta tryptase gets made here, and a gamma tryptase can be made down here. The important place to look, though, is right here. These are 2 places where either alpha or beta tryptase can be made. And the bottom line is actually, in a lot of folks, they just have beta tryptase being made in these 2 places in their DNA on chromosome 16. But some folks, instead of, not some folks, but most folks actually, instead of having 2 betas here, they have 1 beta tryptase and 1 alpha tryptase. So this blue is, let's say, is alpha tryptase. It's in the exact same place on the chromosome as where beta tryptase gets made. And the only difference between these 2 is just a couple of mutations that may have happened thousands of years ago that made it, in some people, it just ends up making a different protein, but a similar protein to beta tryptase. And what happens in folks who have this high tryptase is that, and here's the alpha tryptase. So the point is that the beta tryptase, the red, probably makes beta tryptase floating around in the blood. The alpha tryptase makes alpha tryptase floating around the blood, and that is the tryptase that's being measured when your tryptase is 3.1 or 5.2 or 7.8 or whatever it is. When you have the patients who have the high tryptase, the genetic abnormality that we found was an extra copy of this tryptase that just suddenly appeared in these patients, and we don't know when in evolution this ended up happening, but it was way back. We've never found where the parents don't have it, but the children do. The parents always, at least so far, have had the extra copy, and so we don't know when this happened, but an extra copy was there nonetheless, and we believe it's producing more of the tryptase, and this is why your levels might be higher, like 10, 12, 13, or 15, or something like that. And in a small percentage of the patients we were following, they actually even had a 3rd copy of alpha tryptase in this spot right here, and they had even higher tryptase levels. So again, the genetic reason why these patients have a high tryptase at baseline is that they have extra copies of alpha version of tryptase right here on 1 chromosome. It didn't cause any problems if they had one alpha copy over on one and the other alpha copy on the other. It only caused a problem if there was a second alpha or a third alpha on one chromosome that they inherited either from their mom or their dad. Okay. So then we looked in all of our patients, and we actually made a test that we actually hope soon enough is going to be available for your doctor just to send if it's suspected that you might have this. And we can talk in a moment as to why it would be worth sending this test. We found that there were different ways you could have the two alphas on one allele, and you might have another alpha tryptase even on the chromosome that you inherited from the unaffected parent. Again, that didn't seem to change much, but we did actually notice that in one of our families, and then in more, that both parents, if you notice, both parents are colored in. Both parents had the duplication, and actually one of our most affected patients that we had the duplication on both chromosomes that she got because her parents both carried it, and she was one of our most severely affected ones. And then we also had patients who had the triplications, and we noticed that our patients with the triplications had, and that's such as this patient, had higher levels of tryptase than those who had duplications. So if both parents carried and you had duplications on both chromosomes, your tryptase could be higher, or if you had a triplication, your tryptase could be higher. And in both cases, with this higher tryptase and having either duplications from both parents or a triplication on one allele, on one chromosome, these patients also tended to be sicker than the ones who just had the duplications. So triplications or people with duplications from both parents, they had worse outcomes. So then, but what got us worried is we thought we were studying a pretty rare disease. And what are the odds that a couple would have found each other with high tryptase and a rare duplication or triplication in alpha tryptase? What are the odds that they would have found each other? This got us worried that maybe we were off or something was wrong. And so we actually did is we looked in a couple of databases that we have at the NIH where we have patients who've submitted their blood for DNA analysis and where we've able to save that blood and look for tryptase levels, actually, or anything else you want to look for, but we chose to look for tryptase levels, and then actually we could call the patients back and ask them about their symptoms. And lo and behold, what we found is that every single person who had a high tryptase had either a duplication or a duplication on both chromosomes inherited. They had a duplicate, every single person. So that 5% that has the high tryptase in the general population, it seems like all of them are caused by having the duplications in alpha tryptase. So we were just absolutely astonished that 1 in 20 people is carrying this genetic event of a duplication in alpha tryptase that is associated with having a high tryptase. Now, if you notice, some of these dots, the dotted line that we've drawn here that's 11.4. That's quote-unquote normal. Some of these patients did have a tryptase lower than 11.4 and carried the duplication. And even overlapped with some folks who didn't carry the duplication. So these two patients here had a tryptase of 10, and yet here are others who have the same level of tryptase but have the duplication. And so those are the folks where it really might be worth doing a genetic test to figure out if they were symptomatic, if that's what's causing it. The other thing, of course, to do is just to check the tryptase in your parents or in a sibling or in kids and see if more than one of them has an elevated tryptase, because almost in every case that's caused by the increase in the number of tryptase copies on one chromosome. So we actually were able to then call the patients who had the duplications. When I say patients, I have to clarify. These were, quote, normal volunteers who came in just to have this study done, to have their DNA checked, and to have blood stored and to be called back. These folks were essentially, they weren't referred to the NIH for a rare disease. They were just normal volunteers. In fact, there were plenty of these folks who were employees at the NIH who just wanted to, find out what was going on in their genes, and that was about it. Well, it turned out that when we spoke to 9 of the folks who had the duplications and compared them to those who didn't have the duplications, actually 4 out of the 9 of those patients basically could have been ones who would have walked through our door in the original group with all the same issues of itching and flushing and belly pain and chronic pain. And actually 1 out of those 9 had very clear hypermobile EDS. So that's quite common. Okay, so that means that out of 5% of the population, just only a little bit less than half of them were highly symptomatic, and over 10% of them actually were hypermobile and had all of those symptoms. So that would at least imply that it's possible that this genetic event might cause this type of a thing in the general population. And that was quite amazing to us, but there's much, much more we need to do before we can be sure about this. So the commonly seen elevations that we see in tryptase that are associated with lots of symptoms that can be attributed to mast cell activation, but again, I don't know that it's being caused by mast cell activation. These elevations could be associated with connective tissue problems. The common elevations seem to be almost always caused by increased copies of the alpha tryptase gene, and probably the families that we saw originally were like the tip of the iceberg, and that there are tons of people out there who are on a continuum. And it's clear to us, so 4 out of those 9 had lots of the symptoms we described, but 5 out of those 9 really had absolutely no issues at all. So you could be walking around with a tryptase that's high, and you could have these duplications, and you may have absolutely no symptoms at all, or it could take the stressful event or whatever it is to trigger the symptoms. And then finally, we definitely were referred patients, and this is probably going to be relevant to lots of folks who are listening now. We've definitely been referred patients who have all of the features that I just described, who've got all the things that might be associated with mast cell activation, the dysautonomia, the itching, the flushing, the pain, the connective tissue issues, but don't have a high tryptase, and we've checked them, and they don't have the duplication. And what's causing it in those cases, we don't know. We've done whole exome sequencing on a number of those folks and gotten absolutely nothing valuable out of that yet. It may take a lot more folks to be sequenced, or we may need to try and find some other marker, the way that the tryptase was able to guide us in understanding the people who don't have a high tryptase, have all these symptoms, but don't have a high tryptase, and don't have the genetic duplication. Another important point of what this means is, look, 1 out of 20, it's 5% of the population. That's millions of people in America. And that means there are going to be huge numbers of coincidences. Huge numbers of people are going to have duplications or duplications of alpha tryptase and high tryptase levels, and tons of other problems that are just completely by coincidence. Because if you take millions of people and you have diseases that are just in a fraction of them, that's still lots and lots of people. And so it's critical to point out that there absolutely could be more than one thing going on at the same time. The things that are absolutely being caused by having the extra tryptase allele, that's exactly what we're working on right now, is we're going very carefully from disease diagnosis to disease diagnosis to sequence each patient, for instance, patients who have hypermobile EDS, and patients who have, I'm not even seeing these patients, we're getting them from researchers who have that DNA stored, patients who have irritable bowel syndrome, patients who've had to have their teeth pulled, that sort of a thing, because they didn't fall out when they were kids. Each one of them has to be carefully understood and diagnosed, and then their DNA checked to see, because if it's just 5% of everybody who has IBS who have these duplications, then we've explained nothing, because that's what it is in the general population. And that's the hard part of what we're trying to understand. And another point about where there really could be more than one thing going on, a study was recently done in the New England Journal of Medicine where they had 7,000 people were referred for what looked like a genetic problem. And in 5% of them, there was more than one major genetic diagnosis that was present. It's a coincidence that it's 5%, but the critical point here is that 1 out of 20 people with an incredibly rare disease that deserved to get whole genome sequencing, 1 out of those 20 had a second genetic problem going on as well. And so we should not assume that there's only one explanation for what it is that's going on in these patients or in patients like this. Okay. An interesting point, a point that might be of value to finding what we found, is that, as I told you, at that one spot on the chromosome, you could either have alpha or beta tryptase, and it's just what differentiates the two at that one spot are a couple of mutations in alpha tryptase that not everybody has. And actually that's been quantified. And in fact, 30% of the population across a number of ethnicities, 30% of the population has no copies of alpha tryptase, none at all. Now, we have no idea whether they're sick or not or whether they have issues. We really don't. But the really important point is that means you don't need alpha tryptase. And so if we could find a way to block or get rid of alpha tryptase, that if we can prove that the alpha tryptase is what's causing the disease directly, and it's not just associated, then wow, that could be a rather amazing way to fix that problem. And who knows, it might actually contribute to disease in other people in ways we just don't understand, not just those who have the duplications. But that's obviously a research question that will need to be answered in the future. But the point is that if 30% of the population doesn't have a protein, it's probably okay to not have that protein. Now, who knows why we have alpha tryptase to begin with. If it's in that many people, it must have done something. It may be that it was something that was advantageous. We have noticed that most of the patients who we saw who were referred, who've got the duplications, are from Northern European extraction, although it's not, perfect in that regard. So maybe there was something in Northern Europe that was that having alpha tryptase helped. The duplications, we really have no idea. We have no idea what the duplications might be good for, but anything that's present in 1 in 20 people implies it must have been something important. Okay. So what's next? What is to come up? And what are we going to do moving forward? We've got to figure out how many symptoms are the direct effects of mast cells letting go what's inside them versus a direct effect of having just too much of this tryptase floating around in your body versus what happens when you've been exposed long-term to either what's inside mast cells or the tryptase, such that it no longer matters if your mast cells are active. They've already sort of done their thing, and it wouldn't be changeable. Those are critical questions that we're going to need to ask, and it's going to take a long time for us to really get at it, as frustrating as that might be. Believe me, I'm the first person to say that I wish I had more answers. It drives me absolutely crazy, and it kills me when I don't have answers for what's going on in this very, very frustrating disorder. I'm sure it's of course tremendously frustrating to those who are affected, but to those of us who are really trying to understand it and studying it and to study it, it's deeply frustrating, and we are really motivated to figure out what it is that's going on. It just takes time, and it sure as heck isn't easy, and I that's what I can say. And so, are the changes happening? Are the changes happening to cells? Are changes happening to connective tissue by chronic exposure to the things that are either inside mast cells or to the tryptase? Are there long-term changes to behavioral responses? And by behavioral, I mean things that you have control over and even things you don't have control over. It could also even mean where your heartbeat just starts picking up because it's been conditioned to do that, and it may not even have anything to do with what originally caused the issue. And so what do we do? In the meantime, and so those studies are happening, they're ongoing. I can't say that there are lots of folks out there who are rigorously studying this, unfortunately, and I hope that we can get more and more of our colleagues interested as they see that there are some feet in the door, like this genetics change that we found. It's a foot in the door that might be able to help, and people of different, to be honest, I have no idea how to take care of dysautonomia because I'm not a cardiologist, but we're hopeful that cardiologists or neurocardiologists will pick up on this and start doing research on it so that they can also try to understand this very complicated interplay. They also have diseases that affect dysautonomia but may not look like mast cell activation syndrome, and so they're studying it but maybe not as directly. In the meantime, we treat symptoms. We try to stop mast cells from releasing what's inside them, and we try to block the things that mast cells create. There are, unfortunately, not a ton of medicines that do this. There are absolutely no clinical trials that have been done, randomized, double-blind clinical trials that definitively state that even antihistamines are for sure working. I can only say anecdotally that lots of our patients say that it helps them, but it's completely a trial and error type of a thing. We don't have evidence that any actual medical intervention works. We only have anecdote and experience, and different people, different caretakers, probably depending on the type of patient that gets referred to them, have different experiences with their patients. So it's not even like, well, if I go to the greatest grand poobah in the world, they're going to have an answer for me because I might be different than the ones that they're treating. And so we're desperate for randomized trials to really tell us which of these meds are really working and in what types of people does it work. Can we get a better idea beforehand of what will work, or are we really just going to do trial and error? In the meantime, absolutely positively trial and error is what we say for folks. I've definitely had patients, I'm relaying it, again, it's not in a trial, it hasn't been validated, but patients have said that stress management techniques have been very helpful for them, cognitive retraining, meditation, mindfulness, those types of programs. Sometimes they involve exercise, sometimes they don't. I know that it's certainly a controversial topic in a variety of chronic disorders. I can only relay that there have been folks who have gosh, they've been on SSI disability from this, and they have the duplication. They've got the genetic change, and they've enrolled in some of these programs and actually have completely turned it around. I have other patients who say, forget it, that's never going to work for me, and I just can't say. I don't know, but we've certainly seen that has worked. Of course, we'd love to find out if blocking tryptase is going to cure this, and that's something that we are actively working on right now. And then, again, could this be a foot in the door for those who don't have the genetic duplication but have all of these symptoms and who don't have a high tryptase? Could this be related, like let's say a tryptase receptor like PAR2? Could a mutation in that or something that regulates that be explaining what's going on in other folks? Or is it completely a different cause altogether? These are things which we're going to need to answer. So there are a couple more points I wanted to make. First of all, I just wanted to highlight this is at least a part of my gang. This is Tom DiMaggio, who's an amazing nurse coordinator. Oops, let me try that again. This is Tom DiMaggio, two guys in sunglasses and hats, who's an amazing nurse coordinator who's in my lab, who's really helped us bring in lots of the patients and coordinate. And then John Lyons right here, excuse me, is, again, the person who really spearheaded this in my lab to solve this amazing genetic issue and continues to work on this. A couple of points I wanted to make. Right now, we are not bringing in new patients to the NIH with this because we're trying to focus on understanding what's going on and answer a lot of the questions that I just mentioned. I'm certainly trying to work with partners to see if we can get other folks to start to see if there are genetic explanations for those who don't have the high tryptases. But I only have so many folks. There's only so much that I can do myself, and so it comes rather overwhelming to respond really, and it kills me. It kills me that we can't take care of this more. We really don't treat folks at the NIH for this. We help. We give some expertise. We see if the duplications or the triplications are there. But for the large part, we've really been trying to concentrate on understanding what it is that's going on with this particular genetic issue. And so, for now, we really are not bringing new folks in. Most of the time, what I say to folks is, let me talk to your local allergist and try and explain what it is that's going on so that they can take care of you and perhaps the many other folks in their practice that may have this. And the other part is that I go around the country and the world talking about it so that to try to get folks to understand and see what it is that's going on so that they can become more educated. This really is something that most clinicians do not know as much about because, to be frank, there just isn't that much literature about it that really explains what it is that's going on to really give you a handle on what's going on. And as such, it's not like there's such extraordinary things to do. It's just that same patience and trial and error to try and help with this disorder. You can't. Okay, a word about the actual test. So we're hopeful that very soon there will be a lab that you'll be able to send this test off and get it into your record. Even the test that we do at the NIH can't really go into the official medical record because it's a research test. So that's why we're working with a commercial partner to do it. because it can't be done by regular whole exome or whole genome sequencing. You will not find it. You will not find it with 23andMe or anything like that. It's not there, which is probably more valuable right now is, as I mentioned earlier, to look at other family members. And if their tryptase levels are high, and again, as I showed you, high could be 10 or 10.5 or whatever it is. If multiple family members are up at that level, the chances are good that those folks are carriers. But again, which symptoms are absolutely positively because of those duplications? We think it's the things that we saw in those patients who were referred to us. But again, it could be referral bias. We've seen those studies that showed that people in that allergy practice who had a higher tryptase had more of those symptoms, but plenty of people who didn't have the duplication or didn't have a high tryptase also had symptoms. It just did seem that indeed having a higher tryptase does give you more symptoms. And that gives us, that's why we keep going on, that we do believe that a lot of these things are attributable, but it's not completely proven yet. So those are the points I just wanted to oh, and another point is it could well be that even if you see multiple people in your family with the same types of symptoms, we're not sure that in every case it's genetic. There may be non-genetic causes for lots of the symptoms. Now, hypermobility certainly seems like something that you're born with. It certainly could be, but for all we know, exposure to tryptase in utero could be enough to cause the hypermobility. We do have some patients where the mom had a duplication and had children who don't have the duplication, some of whom were hypermobile. Now, either that's what's going on, that somehow in utero this exposure caused that, or it could be that the hypermobility was totally unrelated. And again, that's just an ascertainment bias as to how the hypermobility may be related. Again, the tryptase does act on the types of tissues that lead to joint formation, as do other mediators from inside the mast cells. They do. But when that happens, is it when you're born? Is it when you're in utero? During growth, it causes one to become hypermobile. We don't know the answer to that, but it is possible. We have these families, and again, or it just could be that two different things are going on, and sometimes the hypermobility is caused by the duplications, and sometimes the hypermobility is caused by something else. The hypermobility that you see, of course, also is in certain populations relatively common. So you have that same problem that the coincidences are going to end up happening, and we don't understand that very well at all. I know I've sounded like a broken record when I say I don't understand. We're doing more research, but to be honest, I'm just being honest. So that's what I have to say. I'm going to go look in the Q&A now. Oh, sorry, go ahead.
Thank you so much. Thank you so much. A heartfelt thank you from our whole patient community for doing this talk for us, and then also for you and your amazing team and all the contribution and progress that you've made so far. We're just very, very thankful for that. And we do have, like you said, some questions from the live audience here. If you have time to take a look at that, the Q&A box should have a list started for you.
So I'll try to go through them. I'll try and screen very, very quickly, but I want to go through them also. Can I explain the link between, so should I just start and read the question? Is that what I should do?
Yeah, that would be super if that works for you.
So can you explain the link between tryptase levels being elevated, mast cell issues, and chronic pain? Are there ways to treat tryptase mast cell that have been shown to reduce pain? So number one, the answer is have been shown to? Nothing. There's no study on where a trial has been done to reduce pain. We can only say anecdotally, and then there have been some observations that blocking mast cells reduce many of the symptoms. Pain, I don't think, is one of the outcomes that was measured in the anecdotal published reports about giving like antihistamines and stuff like that. My daughter does better taking Zyrtec and Zantac, but I don't get how this helps with pain reduction. So it could well be that it helps with pain. As I had said before, blocking histamine or blocking anything that the mast cell is making may acutely prevent the things that histamine does, but it also could be that over time, when histamine acts on a blood vessel or even on a nerve, that over time that changes the way the nerve or the blood vessel communicates. And one of the things that can communicate is pain. And so it could be that chronic exposure to any number of these things can contribute to pain, and blocking it eventually could stop that pain, not that immediately the pain is happening because the mast cell is granulating or something like that. But again, that has not been directly shown to reduce pain. Would any markers for familial high tryptase show up with whole exome sequencing? We've actually looked at this. 2/3 of our families did have a marker that showed up in whole exome sequencing. 1/3 did not. There was no difference in the severity of affectation if you had it or not, and there were plenty of people who did not have the duplication who had the marker. I essentially would say it's not valuable. It wouldn't tell you anything additional, but that's a really a good question. We're very much looking to see if we can find something that is more tightly associated with the duplication so that you wouldn't have to do the test for the duplication and you could just use whole exome sequencing. We'd love to be able to do that. Okay, so so this person is saying that they were tested for VEDS, received a result of genesis did not recognize. I don't know what exactly that is, but this person does feel better on antihistamines, does have autonomic dysfunction. Local doc's not very familiar with EDS. Is there and is there a find a doctor to investigate this? The answer is yes. I'd love for you to find a doctor to investigate this. And the question is also whether this could be due to a mast cell activation issue. If the antihistamines are working, that's great. Is it a mast cell activation issue? I'm not sure. It doesn't much matter for the most part right now because the only things we know are how to give those antihistamines. A change in COL3A1, which is associated with a collagen gene, that's really not at all related to what we're talking about in these patients. Collagen gene changes could be associated with some of these symptoms, but we have not seen that, and that's in our patients who we're following. Their collagen is not being disrupted at all genetically. Other folks who have genetic collagen changes, I have not seen any good mutation by mutation analyses of the symptoms that we're talking about with respect to flushing and itching and that sort of thing. There is some on dysautonomia, but again, I'm not sure mutation by mutation if that's been worked out. Okay. How large was the cohort? Well, the original cohort was 100 meaning at this point in time, I think we've seen 150 or 200 patients who have the duplications. The families coming from about 50 or 60 families. It was about 1,000 patients that we screened in the general population to find that indeed 5% of them had these duplications. It was a smaller number that we could actually reach to actually interview and find out about their whether they were symptomatic. Would there be any reason for hypertryptamine to be associated with mitochondrial disease? No, not at all. They're just not going to be related. It's having the duplication in tryptase is the reason why the tryptase is high. Whether mitochondrial disease might exist at the same time, I think it's very difficult to say. I think we need to know a whole lot better about how mitochondrial diseases might cause these symptoms. I've not seen any evidence that would have anything that it would be related. I know that it's a term that gets tossed around a lot. I don't think it adds much at this point in time. Any cases of eruptive nevi with people with mast cell activation? I have not seen that. Do you check and do whole kit sequencing of patients with suspected mast cell activation disorder to see if they have mutations other than the famous D816V mutations? It has been done, but we don't. We do. We don't see it. We haven't seen it in any of the patients who were referred to us, but it's possible. Are there experimental PAR2 antagonists that could be used to see if tryptase actions are mainly or through PAR2 receptors? They do exist. Again, we don't have. We would have to do it in patients. I don't think they've been given to patients. But pharma has looked at PAR2 antagonists for a variety of different reasons in the past, but haven't gone into patients with them. Unfortunately, we have no mouse model of this yet. We have to make a mouse model to know if it's going to work. Okay, oops. My adrenal glands failed a few years ago. I had 2 years of daily steroids before I was able to wean off of them. I'm not sure what the question is, but it does bring up a good point. We've had plenty of folks tell us that steroids made them feel better. But that is absolutely not a solution, of course, excuse me, because you can't stay on those steroids precisely because it'll make your adrenal glands fail if they haven't already failed, and cause all sorts of other side effects. So staying on them is not. But the important point is, in acute settings, steroids making folks feel better may actually be just as much. It could be anti-inflammatory. I don't know. But also, what steroids can do is suppress stress hormone release, and it's important to keep that in mind. This is just very much this is not clinically relevant yet, but to the extent to which we can stop certain stress hormones from being released, and a steroid does that, that's giving you a hint that that might be what's helping in some folks. A decent number of folks have said it works great, but they cannot tolerate staying on them, and they shouldn't stay on them. When will the genetic test be available commercially? Hopefully soon. Are there people with less than normal tryptase? Great question. There are definitely people running around with zero tryptase, and there's nothing wrong with them, and we've actually seen some because we've been asked that question. Could a tryptase blocker be created? Absolutely. That's what we're working on. Whether that's going to work, different question. Not sure. What are the names of the genes and the SNPs that show the tryptase duplications? Tryptase, TPSAB1 is the tryptase gene. The alpha tryptase gene is T as in Tom, P as in Paul, S as in Sam, A as in aardvark, B as in boy. 1 is the gene that is, when it has the alpha mutation, which is it's actually not very easily detectable, the alpha mutation in TPSAB1, when you have more than one copy on one chromosome, that's the duplication. So again, it would be hard to, you couldn't look that up. There are 60 children in the world with Timothy syndrome, which has a known genetic marker. 3 of their mothers have HEDS. Do you find that's statistically significant? Potentially, I don't know well enough, and you'd have to calculate. I just don't know well enough. And also, you'd have to know the ethnicity and all sorts of stuff like that, but it's a good question. Again, have exome databases such as EXACT been evaluated to check how common the gene variants are among the general population? These will never be in EXACT. You can't find them that way. Only through right now by doing this particular test that we came up with. And again, we'd love to find a marker in EXACT that would correlate, but right now we don't. Is there a correlation between having no or low tryptase and no or low natural killer cells? No, and we've looked. How would a family go about becoming part of my study? Right now, we're not admitting new people into the study, as I mentioned, because there's nothing we could really add right now to what we have. From the patient point of view, we really need to now get at what we're like I said, to look at individual diagnoses with people who have like hundreds of patients with the same diagnoses, and we're checking that DNA, and then at the same time we're doing lots of lab work to try and understand within research bench work to understand how this alpha tryptase is causing the trouble that it is. I gave my daughter an antihistamine and treated her amenorrhea and greatly reduced my daughter's severe POT symptoms surrounding her period. We certainly hear that story, absolutely. And antihistamine stopping pain from inflammation, just always remember that there are different kinds of inflammation. So the inflammation you would take aspirin for is very different, but nonetheless, for some folks, aspirin is helpful. Antihistamines are causing a very different kind of inflammation. Again, it's usually not associated with pain for antihistamine, but who knows what would happen over chronic periods of time. Our pediatrician wanted to order a tryptase level for my kids, but she didn't know how to code for it. Any suggestions I could relay to her? Unfortunately, at the NIH, we have no ICD. We don't use insurance. We don't, because it's all, you're paying for it. Your tax dollars are paying for it. So I don't know the ins and outs of getting insurances to do these things, and but I guarantee you there are folks who do know that. And I'm actually sure that Dr. Maitland will know that next week. I assume UDN, by that the person means the Undiagnosed Disease Network. They're looking at patients coming into the program for elevated tryptase. They usually just call us. So, and then we would say check a tryptase or something like that. So the Undiagnosed Disease Program, it's not magic. It goes to someone who knows about the symptoms that are there. So they often come to us. Does this mean that stress from having EDS can trigger these mast cell symptoms? The answer to that is it is possible, but I do not know for sure. It is possible based on a number of research findings, but it is not proven until we do a trial. But it's something to at least think about. But I would take this opportunity to say as clearly as I possibly can that this is not something that's, quote, in people's heads, unquote. And I'm always a little bit nervous about saying the business about stress and the mast cell activation, A, because it might sound like it's, you know, oh, if you got it together, you wouldn't have the symptoms. We all know that's a bunch of baloney. B, because it might make a doc say, oh, you see, it's all in your head. And so I'm careful about it, but there are a number of reasons biological reasons why it would make sense. And so it should be, I think it's important to pursue it with the caveats that I don't want it to suddenly become this thing where you're saying it's all in your head or something like that. Would you expect gene therapy to silence or downregulate alpha tryptase to alleviate symptoms? It totally could work. I'm just guessing there are probably easier things to do than gene therapy, like making an antibody that just blocks alpha tryptase. That might be a lot easier than gene therapy. Once CRISPR really gets up and running, as you saw in all of the major news outlets, on Monday it was published in Nature that a genetic change could be fixed in an embryo. Who knows how long it'll be before we can do that in walking and talking people. We'll see, but it just might. You may not need to do such a complicated thing as gene therapy. There might be easier ways to deal with it, but it's possible. What do you suggest for those of us who are still struggling to feel better, chronic pain, chronic fatigue, multiple other symptoms with few answers? This is the question that I get asked all the time and constantly. We need folks to do research into these specific symptoms and to try things and or find genetic things that might have an underlying genetic cause. That's the best thing. But in the meantime, it's trial and error. And always remembering that anecdotes don't necessarily mean that that's what is going to work. And really sticking with someone who has patients to work with you, whether that person is your country doc or at a major academic medical center. Can mast cell activation increase hypermobility? As I said, there are things in mast cells, including tryptase, which can act on connective tissue absolutely positively. I had samples taken to the NIH for a different genetic test that were kept. Can I request that tissue be tested for the alpha tryptase abnormality at some point? It might be possible, but we wouldn't be able. It wouldn't be something that we could really report to your medical record because it's a research thing. It would be far more valuable to do the test once it comes out, and it really is not going to be that much longer before that test comes out. I keep saying it, and by the way, I don't make $1 on the thing, which is killing me, but I don't make $1 on the thing. I just assume find some other way for it to be done without it having to be so difficult. Do you have experience with the newly Zizal? How does it compare to fexofenadine? You talk to different people, they'll tell you different things. So some people can take generic but not generic. Some people can take one version of Claritin versus Clarinex, all that sort of stuff. No rhyme or reason that we have seen. Again, that's anecdotally. Have we explored whether early exposure to antibiotics may be a factor? It's too hard to do that in the numbers that we've been following to really know. The folks who have the duplications, whether that makes them sicker or not, with only 150 folks exposed to antibiotics, it would be hard to really do that. And these people are 30 and 40 years old, so it's really, really hard. We have kids, but now it's even fewer that are kids, where we would really remember. That would take a large, what we would call a prospective cohort, where you take a lot of people and follow them over time for lots of things, and that could be one of the things we could tap into. Can the additives or preservatives in our foods that we have been exposed to, especially since the 80s, contribute to the increase in MCAS? That's possible, but again, MCAS is perhaps 20 different things. So there may be one form of it where it's being changed, but there may be tons of others where it has absolutely no effect whatsoever. I have not noticed, in folks who were more organic versus not, that there's any difference in the severity of their disease. Is there an increase in MCAS? Is it seen more in a specific generation? That's a great question. There have been a paper or two which have suggested that maybe it has gotten worse, but you don't know if that's because of something environmental or if that is a genetic event, just like the changes in our microbiome are partially environmental, but they're partially just drift over time, and those are actually changing things in our genes even to some extent. They don't change the code itself, but how the code gets expressed. Those things can happen over time, and it might be hard to know what's contributing to it. Whether it impacts our folks with the 2 copies of tryptase, I don't really know. I would say that it's not like we've noticed, like in the parents' generation, when we're looking, we've seen 1, 2, and sometimes 3-generation families. There hasn't been a big difference between grandma, mom, and the kid. In some cases, the kid's sicker. In some cases, grandma's sicker. So there's not been a clear pattern. Have I seen co-occurring immune deficiency problems in this cohort, whether or not having high tryptase? Very, very little. We've seen in a decent number of folks fevers, which is interesting, but not where there's no cause. Fevers, and some folks have had some mouth ulcers, and the mouth ulcers were again we have not, and so we've seen that, but not actual, not an increase in infections per se that would make you think of an immune deficiency. What was the highest, but again, I have seen immune deficient patients where we know the gene and it's rare and there are 30 people in the world with it, and 2 of them have even hypermobility and mast cell activation. So go figure. What is the highest tryptase we saw studying families with it not being mastocytosis? Baseline tryptase of 22. Mother has a baseline tryptase of 49. You do not win the prize. We've seen it higher, but that's a great question. And that would imply a triplication or more in your family. That's a great question. Can the doctor in the DC area call my office? Yes, they can. When it becomes such that I can't get my work done and talk to the docs, then we'll have to figure out a different solution there. Does tryptase and mast cell, and I assume mast cell activation is what the person means, cause food sensitivities? So interestingly, sensitivity. So I wouldn't say that there were more classic food allergies in our patients with the higher tryptase, but being highly reactive and in many different ways having nothing to do with IgE or true allergy, that we certainly saw in our folks with the higher tryptase. But I don't know, again, is the tryptase causing that? I don't know. Is the NIH sponsoring genome testing for tryptosemia? Well, yeah, that's how they're paying me. For hyper-IgE, of course, that's what I do. So this is just one of the diseases I study. We've studied quite a number of other hyper-IgE syndromes and try to find the genetic causes of those as well, and those studies are ongoing and they fund me. There are other folks who study hyper-IgE as well. I'm not aware of anybody else doing NIH-sponsored research on alpha-tryptosemia right now. I would love for them to join the party. Great question. Would Prilosec be similar to Zantac or Zyrtec? No, absolutely not. So Prilosec is a proton pump inhibitor. It does work as an antacid, but it's not an antihistamine, the way that ranitidine or Zantac works as an antacid and as an antihistamine, and that's a really important point. So it's not that you need to be on an antihistamine, you need to be on Zantac, ranitidine, and there are a couple others like it, H2 inhibitors, and Zyrtec is an H1 inhibitor, which is not an not an antacid, but it is a pure antihistamine. A lot of folks who we follow complain of GERD-like symptoms, reflux-like symptoms, and whether the Zantac is working as an antihistamine or those symptoms are happening because of the response to too much histamine, and so when you block with Zantac and Zyrtec, you feel a little bit better. That's certainly something that it could be one or the other. Okay. With the duplication of alpha tryptase with baseline of 11.4 being a fairly new discovery, are the doctors who are renowned for treating mast cell familiar with my study? Yes. Also, what's the benefit of taking the blood test that our family doctors could order that you mentioned in your talk? So to be honest with you, right now, I don't know if that's what's causing what's going on in your family, or at least may be causing what's going on in your family. The other the thing would be is if you have a concern for mastocytosis, and you have this is something that we're working on, it would be good to know if you have the duplication, because that makes it much less likely that you're going to have mastocytosis if you have the duplication in this gene. That's something that we're actively working on right now, is to see could we avoid people getting a bone marrow biopsy if we find that the genetic duplication is there. Again, for right now, a lot of people just want to know. They want to know if that's what's going on. Also, when it's borderline, when the tryptase is 9, 10, or 11.3 as it were, then it's, you don't necessarily know, or even 11.5, is it really the duplication there, or is that just not at all related? So that in the future, should something come up that specifically treats the duplications, you'd be able to say, I already know. And that might be another way that could be helpful. What are the psychological impacts you have seen? And could some cause of ADHD be misdiagnoses of high tryptase? Totally could be. We've seen a huge array of psychosocial symptoms. Again, I have to caveat by saying I don't know how many are being caused, but certainly associated with these duplications. We've certainly seen autism spectrum, absolutely positively. We've seen plenty of ADHD. We've seen some OCD. We've seen tons of people who've been told that they have anxiety and depression, but I'm just not sure how much of that is anxiety and depression, how much of that is a response to having all of these symptoms all the time, and how much of that is something specific and that is being caused by either the release of the mast cells or the response to tryptase and other things that are inside of mast cells. It's a great question. I've been trying to determine if extremely dry mouth is an effect of the antihistamines, or is it possible that extreme dry mouth is a result of mast cell activation? I doubt the mast cell activation is causing extreme dry mouth. We do know that dry mouth antihistamines certainly can cause a dry mouth. Are you saying that you're seeing patients? Yes, okay. Are we only seeing these symptoms in hypermobile EDS and not necessarily in the EDS that have gene tests? That is precisely what I'm saying. We have not really seen these symptoms as much in folks who have other EDS diagnoses, although, again, I don't know how carefully each one of those genetic ones has been looked for these types of symptoms that we're talking about that are been associated with mast cell activation. I know dysautonomia has been looked at carefully, but the rest I'm not so sure, and so one would have to wait to be seen. Every one of the patients who we saw was hypermobile EDS. Those are the ones who were referred to us, the ones who have the duplication. Every one of them who had EDS, it was hypermobile EDS that we're talking about. The name of the test for determining tryptase levels is a basal serum tryptase. Actually, it's just a serum tryptase. Basal just means it's not when you're having, like you just got a bee sting and you anaphylaxed, or you ate a peanut and you anaphylaxed. If you're symptomatic, it's okay. We tend to want to see what it is at your baseline, whatever your baseline is, and not as much in a flare. That can give us the best idea as to what it is. So we say basal, but when the thing is ordered, it's just called a serum tryptase. How do you get an accurate tryptase level? Like I said, the best time is just, and who does it? Any doctor can order it as long as they know how to order it. And again, I'd have to defer to those who are in the community for the best way to order it and get it paid for. But to get an accurate, it's not a matter of accurate, it's just a matter of sending it. The Mayo Clinic and Virginia Commonwealth University are the ones that have the ones that most folks use when they send off that clinical test. But it really doesn't matter. If you're on an antihistamine, it does absolutely nothing to the you can be on any histamine you want, and it will not change your tryptase level. But if you've just had anaphylaxis and you get it drawn, it may be high, and you may want to draw it again when you're back down to your normal level. That's an important point. Any idea what it is in artificial dye that seems to be a huge degranulator in severely affected population? I don't really know. I'm sorry, I just don't know the answer to that. Have I sequenced this locus with long reads? Like long reads means going through long stretches of DNA, as opposed to the way that most whole exome sequencing is done is 100 base pairs at a time. Just to give you an idea, what we're looking at is a 40,000 base pair stretch of DNA. We need to go all the way across that to really know exactly what we're looking at. That's something that we're doing on a research basis right now. Do you ever see, to use these other ones, use the other types of technologies, do we ever see partial duplications of alpha tryptase that might affect detection in the bloodstream with common tests? No, not really. Is it possible that trifecta, so patients without elevated alpha tryptase have copy number variants in other tryptase genes? We've looked, we haven't seen that. Great point, and maybe we just haven't looked the best way, but we can use our test to look for beta and gamma tryptase duplications, and we haven't seen that. But that's a great question. I wish that were the answer. Boy, do I wish that were the answer. And just for the trifecta, for the For those who don't know, that's often is EDS, POTS, and mast cell activation syndrome. But to be honest, for me, it's the 20-fecta. It's lots and lots of different things that folks have. How long will it be for this research is available to our doctors? So there is absolutely positively a paper that we published already in Nature Genetics in October of 2016 that has most of the results of what I just showed you just now. We also then put a FAQ on the NIAID website. If you just Google FAQ and hereditary alpha tryptosemia, you will find a pretty decent FAQ that I would show to my doc. There's also a press release with a video interviewing me. I shaved that day so that, to do a video that might help you as well that is linked to it. But if you Google hereditary alpha tryptosemia, which is what we're calling it's a mouthful. Maybe some folks could just call it alpha T. But hereditary alpha tryptosemia, if you Google that, you will see from the NIAID the press release and a video about it, and then also the FAQ, which you could show your doc if that is helpful. Could a high tryptase, 27, cause a high IgE, 2200? I don't know that it causes it, but we certainly did see in a small subset of the folks who had the high tryptase, they also had very high IgE. If multiple folks in the family also have a high IgE in the thousands, that might be something that we'd want to look into because it may well be that those are happening as coincidences. Or if they are linked, we'd want to understand that more. We had a couple of families where the IgE was pretty sky high along with their tryptase being high. I am diagnosed with, this is going to be a tricky one. I'm diagnosed with musculocontractural EDS with the corresponding mutations and also have the D816V KIT mutation and all of the 15 symptoms you delineated, but I do not have a high tryptase. Do I have mastocytosis? So the D816V mutation usually gets detected in your bone marrow or using a really special test, actually one of the tests that we've adopted to look at the tryptase, a special test called a digital droplet PCR to look for D816V KIT mutations because they're not easily detectable in the blood. You usually will not see that by whole exome sequencing or something like that. The diagnosis of mastocytosis would have to do much more with what the bone marrow biopsy showed and if it was evaluated by an expert in mastocytosis. I am not an expert in mastocytosis, but for sure to diagnose that, you would want someone to look at that bone marrow biopsy. That's much more important than the high tryptase. Having the D816V is a major part of that diagnosis. But that is, I don't think, it would depend if you have the D816V mutation, if you were born with it, could that lead to musculocontractile EDS? I don't think so, and as you said, you have the corresponding mutation, so that may be an example of where there are two things that are going on that are not related. If a patient is symptomatic with low tryptase and high IgE, symptoms improve with antihistamines, how does significantly elevated IgE circulating in the body affect mast cells? So it doesn't really. It's just that having that high IgE, having any IgE affects mast cells. Higher or lower, we don't see patients being worse or better with higher or lower IgE. It's usually reflecting something that's going on underlying, but lowering the IgE level usually doesn't help, but whereas blocking IgE for anybody, whether they have low or high IgE, can be helpful, and that's the drug omalizumab. That can help specifically for severe asthma and for urticaria, and in some of our patients for some of their itching and flushing. Is there a correlation between alpha tryptase and primary immune deficiency? We've really not seen that at all. But remember how to define primary immune deficiency as being recurrent infections from birth, usually that are caused by a specific genetic disease. You may find someone with a primary immune deficiency who also has the duplication in alpha tryptase. Is there a registry that we can sign up for future research projects? I would love to establish that. I need more folks to help with that. I need more researchers to be doing that so that it can become more sort of across the board. If a person has a reaction to Xolair, how does it relate to MCAS? The answer is who knows? We just don't know. We don't know what predicts a person having a reaction to Xolair. People with mast activation syndrome or the symptoms I've described have reactions to all sorts of medications, up, down, left, and right. I have a colleague who has done studies, interestingly, that when you're reactive to 4 or more medications, and when a double-blinded study happens, they don't react, and so there may be weird things where, to get back to that whole thing where when the body sees a change that many folks wouldn't even recognize at all, but becomes very sensitive to it, then it might react to essentially the concern that there might be a change happening to protect itself. But that's pure conjecture at this point in time. Are there other than normal H1 and H2 mast cell stabilizers and aspirin, is there any other known therapy that will stop the severe and chronic hot flushing associated with mast cell activation syndrome? Again, first of all, we have to prove that it's the mast cell activation that's causing that flushing. That's very, very important. And we don't know for sure. It could be, really there you would want to make sure that the flushing isn't being caused by something else. It may be harder to prove that it is being caused by the mast cell activation. Again, actually Xolair, which I just mentioned, or omalizumab, that's more of a very expensive and significant thing to take that some folks have done better on. And again, some folks, like I said, on a short term have responded to steroids. Lots of people have proposed lots of different things, but there is no definitive thing, even, that has been proven to help any of the symptoms, flushing included. Do I recommend any supplements that block histamine? By dietary supplements, we just haven't tested any of them. Can taking too many antihistamines worsen MCAS symptoms as well? I have not seen folks who have said that yet. That's about all I could say about that. Does tryptase need to be drawn a certain way, kept cold? No, it just needs to be sent to the right place. Again, Mayo Clinic or VCU do it well. That's just who we send it to, but there are others. Is there association or a link between MCAS dysautonomia and Sjogren's syndrome? Not one that's been reported yet. It wouldn't surprise me, but again, it would have to be proven that it's not just a chance association, and we'd have to be careful about how we know that it's mast cell activation syndrome. Is the erythrocyte sedimentation rate, SED rate, or ESR related to tryptase? I don't think so at all. I've never seen anybody with I have not seen hardly any of our patients with a high SED rate who had a high tryptase. We've seen other folks who do. They've got much more complicated issues than mast cell activation going on. Have you seen the duplication in infants? The duplication is always in an infant, and then when they grow up and become an adult. This is not something that happens during your lifetime. The duplication has probably existed for at least hundreds of years. So it's a genetic event. It's inherited. It's in 5% of the population. You're born with it, absolutely, positively. Symptoms in infants, yes. We've seen some folks who have symptoms in their infants. They've observed them in their infants. Have you seen Dekram's disease or other lymphatic diseases associated with your study? No, we have not. Oh, goodness. Are there ways you can suggest to avoid sensitivities to additives and ranitidine and other over-the-counters? Docs keep suggesting it, but it seems to cause anal cramps. Maybe the red color additives or fillers. I couldn't comment on it. It's so trial and error with folks, I just couldn't say. Are daily low-grade fevers common in those in your study? No, but we absolutely positively have seen folks who get low-grade fevers frequently. Some folks grew out of them when they were younger. Some folks are, they're teenagers or whatever. We have seen that greater than you would expect to see in the general population. Absolutely. What's causing that, we don't know, but that's definitely been an association. And that may not be a referral bias because the folks were not being referred for fevers. They were being referred for, allergic issues much more. Have I seen any patients with any positive Crohn's disease biopsies? I don't think we have. But others may have. So multiple family members with high tryptase probably means we are looking at MCAS and not mastocytosis. So again, I would not call it MCAS. I would call it hereditary alpha-tryptopenia, because I don't know that this is a mast cell activation problem that's causing the high tryptase and the symptoms. There is a single report in the literature of a family that had high tryptase. It came out right around when we first saw ours. One of those family members had, it wasn't quite full-blown mastocytosis, but it was a clonal mast cell problem. But they also had other problems like an enlarged spleen, I think, enlarged spleen and or liver, which is what would make you worried more about mastocytosis. But otherwise, not a single, the others we have not seen that. It just makes mastocytosis substantially less likely if you have the tryptase running in the family. But there are symptoms of mastocytosis that one should always be worried about, and the particular ones would be where you have the urticaria pigmentosa, or that rash on your body, where you would have an enlarged liver, spleen, or lymph nodes, or where you would have like anemia or a low, very low white blood count. That can be of concern that the mastocytosis is causing a problem. Other than that, it would be, you would be much less likely to assume mastocytosis in the absence of those symptoms if you have multiple family members with high tryptase. I'm not sure I understand this question. I'm going to skip over that one. Is idiopathic angioedema the same as mast cell activation syndrome? No, I would really say no. Again, mast cell activation syndrome is a name that's attributed to lots and lots of different things, and it may be manifest in different ways. We really don't have a great sense of that. Either way, idiopathic angioedema can be caused by, first of all, idiopathic means we don't know the cause. But angioedema, in other cases, can be caused completely independently of mast cells. And so it's possible that there are plenty of cases of idiopathic angioedema that do not have anything to do with mast cells, but it also could. If flushing was not caused by mast cell activation, would antihistamine still improve it? Again, it may not be that the mast cell is activating too much. It may be that your body is responding to it too much, in which case your mast cell is just sitting there doing its thing, and an antihistamine could help to improve it. And again, there'd be no test for that. It's just taking the medication and see if you do better. What is the advantage? What advantage is there to doing the 24-hour urine test? This is something that Dr. Maitland may address more. It's one of the ways that the proposed diagnostic criteria for mast cell activation syndrome, 24-hour urine, that's quite a thing to go through. I think it's open for debate as to whether we learn more about what to do for you if it's higher and if it's not mastocytosis. How would someone get their entire genome sequenced? That is an evolving part of the American healthcare system. Do I consider 23andMe a valuable resource for this? Absolutely, positively not. For most things that you could be concerned about, I don't know how it would contribute. I assume that 5 to 10 years from now, everyone's going to have their entire genome sequenced because the price hopefully is going to drop down to right around where your blood count gets to be. And in the meantime, it's a struggle to determine how much whole genome sequencing should contribute to regular health care. That's evolving right in front of our eyes right now, in both the clinical and the research doing this side by side. Is fatty liver disease correlated with mast cell activation? I've not seen that. Have you seen any patients with positive CD2 and CD25 biopsies? Those are markers that you would see on a mast cell. It has happened in rare cases in those with alpha-tryptosemia, but very few. So many doctors tell patients they can't have MCAS with a normal tryptase. Which mediators would you suggest they test for to diagnose? My particular bias is that the testing, beyond the tryptase, where we know it might be associated with a duplication and it may mean something categorically in the future, the other mediators may be helpful to say that might be contributing to what's going on, but in the end, it's the response to therapy that really is going to tell you if something's happening. And for many folks, just trying the antihistamines, H1 and H2 blockade, gives you a hint that it's helping. Whether a test would justify a far more interventional thing, at the moment there aren't too many interventional things that are justifiable with the test. And so that's something that I think different docs would have different opinions about. The question is, what does the label do for you in the end? That's the question. Is there a way to be tested now by our family doctor for hereditary alpha-tryptosemia? The answer is we're hopeful that very soon it will become commercially available by an outside vendor. Is mastocytosis considered cancer? No, but there are people with mastocytosis who can evolve into a mast cell leukemia. Is chemo a treatment? In people who have bad mastocytosis, even though it's not cancer, there are chemotherapeutic reagents that can be used in mastocytosis, but that's in a very severe case. That is really not, again, very relevant to those who have the alpha-tryptosemia. But indeed, when someone has, and again, the chemo would be given for not really for the mast cell activation parts of the mast cell leukemia, but just like any other leukemia where it's taking over your bone marrow and making your blood cell counts go down. That's the type of patient that tends to get chemo. It's really when they start to get, enlarged liver and spleen where you really start to worry about more severe disease. Those tend to be the ones who get chemo or chemo-like drugs. Would tryptosemia cause failure to thrive? We certainly have seen patients with tryptosemia who had failure to thrive when they were younger. Absolutely, positively. We've seen that in folks when they were younger. A lot of folks have difficulty swallowing and had some eosinophilic esophagitis at the same time. Whether that's referral bias or not, we just don't know yet, but it's certainly something that we want to find out. Okay. What about the MCAS people that are still very sick, very symptomatic, failing treatments? What to do in the most severe cases? It kills me. I don't have another great answer for you. It's just working and working with your doc to keep trying, and when your doc gets exasperated, have your doc ask their expert that they tend to go to, and it may not always necessarily be an allergist. It could be a hematologist. It could be a cardiologist who deals with dysautonomia. It's really going after the symptoms. And again, it may not be mast cell activation. It's just an important thing to point out, and unfortunately we don't have great ways of knowing if it is or if it isn't. Is there a protocol for a person with mast cell activation with severe reactions that is different than anaphylaxis? There isn't a particular one right now at the NIH. There may be out there in other places. Has any patient ever had a bone marrow transplant for severe tryptase or mast cell disease? I don't know of anyone. The alpha-tryptosemia, no, I wouldn't recommend that at all. For for mast cell activation, I have not heard of anyone getting a bone marrow transplant. Again, partially because we still can't be sure that it's the mast cell that's causing the problem, and then you go to a bone marrow transplant and it's something else, that would be bad. Can it cause low iron, calcium, and vitamin B levels? We haven't seen that per se in our patients. Okay, I think that's it for the questions.
Wonderful. You made it through quite a few there. Well, people that are here at the live show here are saying you're amazing. Virtual applause and all kinds of other things. So. Everybody really enjoyed your presentation and found a lot of value in it. So thank you so much for your time and sharing your progress and everything you've figured out so far.
Thanks for listening, and again, sorry that I just don't have more answers.
Well, that's the nature of it. Well, we are just really pleased to have you, and we welcome you back anytime, of course.
Okay.
And we wanted to remind everyone that the next session is going to continue on a similar topic here, and we're going to talk about mast cell activation with Dr. Maitland. So please join us for that. That'll be on September 19. And I'm going to go ahead and turn it over to John.
Well, I appreciate, too, all of you attending this evening, and Dr. Milner. This is a really important topic for many of our members. We heard a lot of I got a lot of feedback from people that were really excited about this program. So, and again, we'll have Dr. Maitland on, and this will be the third time she'll come back, and we'll continue to explore this area of mast cell, which is a very important aspect of those of us with that are affected by EDS. So again, thank you for all who have joined. A reminder that these programs are free. Go out and look at our little store, Body Sports store, or if you'd like to make donations, you're free to do that. These programs are totally free, and we want to make sure that people get the information that is available as soon as it's available. So again, I'm going to wrap up for the evening, and thank you for all who have joined. So good night.
Have a great night.